Welcome, and thank you for reviewing LogiX Biosciences. I am presenting our lead clinical asset, Engine 2. Engine 2 is a formulation upgrade of fluticasone propionate. Fluticasone is a synthetic corticosteroid widely used to treat nasal inflammation. This data visualization highlights our commercial objective, the $29 billion respiratory steroid market. We are currently raising a $5.0 million priced round to advance this asset. Our cap table is exceptionally clean. Founders and insiders currently retain roughly 89% control of the company. This structural position is anchored by early institutional validation from the Mayo Clinic. This lean equity structure provides the leverage to execute a near-term M&A strategy with a big pharma partner from a position of strength, rather than operational desperation. To understand the market gap, we must look at the mechanical burden on the human airway, processing up to 15,000 liters of air daily. In chronic rhinitis, continuous antigen load triggers severe inflammation, turning the mucus blanket hyperviscous. This results in mucociliary stasis, a state where the natural clearance of the nose ceases. This heavy mucus physically crushes and paralyzes the nasal cilia. Cilia are the microscopic hair-like structures that sweep debris out of the airway. When they stop, foreign antigens are trapped directly against the mucosal tissue. Standard intranasal corticosteroids successfully suppress inflammation, but lack a biophysical mechanism to clear these trapped antigens. Furthermore, legacy formulations rely on cytotoxic preservatives like benzalkonium chloride and phenylethyl alcohol to extend shelf life. Clinical literature confirms these chemicals paralyze cilia, desiccate tissue, and cause localized burning and apostoxis, the clinical term for a nosebleed. This adverse sensory profile drives massive patient discontinuation. Once the spray is stopped, the antigen load immediately rebuilds, restarting the chronic disease loop. Engine 2 was designed specifically to break that cycle. We combine a reduced dose of fluticasone with our proprietary bioactive suspension vehicle, Lepto-242. Lepto-242 is naturally hyperosmolar. Upon application, it generates an immediate osmotic gradient. This gradient draws intracellular water into the nasal lumen. This influx of water physically floats the hyperviscous mucus blanket, liberating the cilia to resume their power stroke and flush trapped allergens from the airway. By eliminating cytotoxic preservatives and using a citrate buffer to lock the pH at 4.5, we match the nose's physiological resting state. Replacing the chemical burning of legacy sprays with a highly tolerable sensory profile drives the daily compliance required to break the chronic disease loop. This specific biophysical mechanism dictates our regulatory strategy. Because of how the vehicle operates, we can utilize a precise FDA classification. We strictly classify Lepto-242 as a functional mechanical excipient, rather than an active pharmacological ingredient. This mechanical classification explicitly bypasses the restrictive rules governing fixed-dose combination drugs, which usually require proving the independent contribution of each active component. Avoiding those rules secures our place within the accelerated FDA 505(b)(2) pathway. The 505(b)(2) pathway allows a sponsor to rely on existing safety data from a previously approved drug to speed up approval. By manipulating the physical chemistry of the vehicle, LogiX neutralizes the standard regulatory delays that typically hinder biotech development. The next major diligence hurdle is commercial scale-up, or CMC, for complex biologic matrices. Commercial scale-up often fails because standard gamma irradiation destroys the biological profile of natural matrices. Furthermore, attempting to push highly viscous fluids through standard pressure filtration simply clogs commercial pharmaceutical machinery. LogiX has solved this bottleneck through a proprietary aseptic manufacturing process centered on a pre-filtration viscosity reduction step. By integrating a patented additive, we temporarily reduce the fluid's viscosity. This allows the matrix to undergo progressive cascade filtration to physically shed its bioburden. Bioburden is the quantity of viable microorganisms present on a product before sterilization. The fluid passes through standard 0.22 micron sterile filters without clogging machinery. This process achieves USP 61 and 62 compliance at less than 10 CFU per gram using standard equipment. CFU stands for colony-forming units, a standard metric used to estimate the number of viable bacterial or fungal cells in a sample. The topical safety of this matrix is secured by over a decade of peer-reviewed human data. This includes a 42-patient randomized controlled trial at the University of Washington, demonstrating 0 severe adverse events. This graph details the allocation of the $5 million raise. The first $1.5 million explicitly funds our IND bridge, covering the GLP toxicology study and clinical GMP batch. The remaining $3.5 million acts as a strategic multiplier to execute Phase 2 bridging trials entirely in-house. This funding strategy builds the internal capacity to reach Phase 2 milestones, ensuring we dictate the terms of future partnerships from a position of strength. Engine 2 is a direct lifecycle management acquisition target for major pharmaceutical companies. It provides Big Pharma a mechanism to transition their existing respiratory patients to a next-generation formulation and quickly renew their expiring patent exclusivity. This timeline charts our 2 liquidity windows. The first opens at IND clearance in roughly 6 months. The second window opens following the Phase 2 clinical data readout. Based on 505(2) precedents, clinical de-risking at these milestones aligns the asset with substantial upfront licensing multiples and downstream payouts. Engine 2 is mechanically de-risked. It is highly scalable and leveraged for high multiple exits. We invite you to review our scientific blueprint and regulatory filings within our secure data room. Thank you for your time. We look forward to discussing how your fund can lead this round.